Hello, a pleasant morning, afternoon, evening, or nighttime to all BSMT third year sections A through I. I am Miss Merle, and this time I'll be the one to discuss with you the introduction to immune responses, the immune system. However, before jumping directly talking about the cells of that system, we need first to understand the concepts, how the lymphatic system is related or associated to the immune system. Hence, my talk will begin with the aspects of lymphatics and its system as a whole. You might ask, what are the processes that link the lymphatics and the immune system? Oh, well, the lymphatic system for most people is associated with the immune system to such a degree that the two systems are virtually indistinguishable. The lymphatic system is part of the immune system. It is an extensive drainage network that helps keep body fluid levels in balance and defends the body against infections. It is made up of a network of lymphatic vessels that carry lymph, which is a clear, watery fluid. It is a network of very small tubes or vessels that drain lymph from all over the body. The major parts of the lymph tissue are located in the bone marrow, lymph, thymus, lymph nodes, and the tonsils. And also, if you are wondering what this subject really is, we have immunology and serology. So, immunology is the study of the body's defense against infection. And we are continually exposed to microorganisms, many of which cause disease and yet become ill only rarely. How does the body defend itself? When infection does occur, how does the body eliminate the invader, the antigen or pathogens, and then cure itself? And why do we develop long-lasting immunity to many infectious diseases encountered once and overcome? These are the questions addressed by immunology, which we study, or we will study really, in block 1 and 2 to understand our body's defenses against infection at the cellular and molecular levels. So the lymphatic system helps maintain internal stability or homeostasis by adjusting to conditions that are optimal for survival. If homeostasis is successful, life continues. If unsuccessful, disaster or death ensues. So again, the lymphatic system contributes to internal body balance or stability. And despite constant exposure to a variety of pathogens, so many pathogens in our environment, internal and external environment, still most people remain healthy. And thank goodness it is because of our immune system, specifically the first lines of defense. And immunology, we can define it as, again, the study of the host reactions when foreign substances are introduced in the body. Foreign or anything antigenic, non-self, right? Non-self. So the science of immunology arose from the knowledge that those who survived one of the common infectious diseases of the past rarely contracted the disease again. And as early as 430 BC, during the plague in Athens, Thucydides discovered that individuals who had previously contracted the disease recovered and recognized their immune status, and he recognized their immune status of those individuals exposed to the disease. But many of the books discuss about this one. Beginning about 1000 AD, the Chinese practiced a form of immunization known as variolation. By inhaling dried powders derived from the crust of smallpox lesions. In the 15th century, powdered smallpox crusts were inserted with a pin into the skin. When this practice became popular in England, it was discouraged at first, partly because the practice of inoculation occasionally killed or disfigured the patient. And the beginning of immunology as a science is usually attributed to Jenner. So thank you, Jenner. Not Kendall Jenner, but Edward Jenner, okay? Attributed to Edward Jenner for his work in the late 18th century. He was able to observe that a relatively mild cowpox or vaccinia, not the cow but the disease, seemed to confer protection against the often fatal disease of smallpox, and this is known as cross immunity. He demonstrated that inoculation with cowpox protected the recipient against smallpox, again cross immunity. So that's the thing for that. Cross immunity. Okay. His scientific proof relied on the deliberate exposure or intentional exposure of the inoculated individual to infectious smallpox material 2 months after inoculation. This scientific test was his original contribution to science. Jenner called the procedure vaccination, from the Latin word vacca, meaning cow. Vacca, cow, okay? This term is still used to describe the inoculation of healthy individuals with weakened or attenuated strains of disease-causing agents in order to provide protection from disease. And what else? There was also this Russian immunologist named LYMPHATIC who discovered phagocytosis. He was able to see that many organisms, again, LYMPHATIC, many organisms could be engulfed and digested by phagocytic cells, which thus provide defenses against infection that are non-specific. Whereas these cells, which Michnikov called macrophages, are always present and ready to act. Please remember that adaptive immunity requires time to develop but is highly specific. Moreover, during the mid to the late 19th century, Louis Pasteur demonstrated that microorganisms cause disease and discovered how to make vaccines from weakened or attenuated microbes. So Louis Pasteur was the one who discovered vaccine attenuation. So the word attenuation is for him. Vaccine attenuation. Forgive my penmanship. His contribution to science is, again, the principle of vaccine attenuation. But the one who discovered vaccination was Jenner. And thus, going back to Louis Pasteur, thus he was generally considered to be the father of immunology, Please do check check page 2 of Darjan's Immunology, Serology, and Laboratory Medicine. It's in there. So we need to define the words, and in IS we need to know what is immunity, what is susceptibility, the two types of immunity, these are the basic things. So let's start with immunity. Or immunity, according to Tortura, it is or resistance is the ability to ward off damage or disease through our defenses. And the opposite of that is susceptibility. This one is not. So we have being vulnerable, so you're susceptible to diseases, and if you're resistant, your immune system is functioning, so it wards off infection. And other books, IS books, we have Seeley. I know this one is an anatomy book. We have Stevens and Harmaning, a blood banking book. They defined immunity based on the following. According to Seeley, it is the ability to resist damage from foreign substances, including chemicals. According to Stevens, very straightforward, so defined immunity as the condition of being resistant to infection. While Harmaning, this one is a bit lengthy. In the book, it says that immunity is the process by which the host organism protects itself from attacks by external and internal agents. And immunity confers protection from non-self. Non-self meaning not coming from the body, and abnormal self elements. And then we have the two general types of immunity. We have the innate and the adaptive. So for innate, innate is non-specific. It is present at birth, inborn, natural, always available, and provide or gives very rapid responses as compared to the adaptive immunity. And the thing with this one is the recognition of the antigens is non-specific, so it does not involve specific recognition. Specificity and memory are the things that really define adaptive immunity, and not with the innate immunity. So that's it. And please remember that the innate immune system is the body's first line of defense against germs or microbes entering the body. It responds in the same way to all germs and foreign substances. So similar response, no matter if the microbes will vary, which is why sometimes it is referred to as the non-specific immune system. That's why non-specific, again, innate, non-specific, natural, inborn. So that's it. Next, we have the, you might ask me, Miss, what are the cells that are the functional units of the immune system? The effector cells of our immune system are largely pointed to these cells, the lymphocytes. In human adults, lymphocytes make up roughly 20 to 40% of the total number of the white blood cells or WBCs. They are found in the circulation and are also concentrated in central lymphoid organs, primary lymphoid organs and tissues such as the spleen, tonsils, and lymph nodes, where the initial immune response is likely to occur. And the thing with lymphocytes, let's talk about histology first. The cytoplasm is scanty, appearing as a rim around the nucleus. Please take note, this one. And the cytoplasm generally stains a pale robin's egg blue as it's slightly basophilic. So again, the cytoplasm, pale robin's egg blue appearance. And lymphocytes, they are the smallest of the WBCs or leukocytes. And we have two types, general types, T and B, very easy. T lymphocytes that mature in the thymus, and the B that matures in the red bone marrow. So red bone marrow. So that's it. The lymphatic system, its structure and function is consists or composed of the fluid, the clear fluid, interstitial fluid known as lymph, and we have the primary and the secondary lymphoid organs too. And then that fluid moving within the lymphatic vessels is known as lymph. And the major difference between interstitial fluid and lymph is the location. Interstitial fluid, it is found between cells, and lymph if it is located within lymphatic vessels and lymphatic tissue. The lymphatic tissue is a specialized form of reticular connective tissue that contains large numbers of lymphocytes. Functions of the lymphatic system include fluid balance, fat absorption, and immune defense. In maintaining fluid balance, the lymphatic system prevents edema. It prevents edema. As the interstitial fluid begins to accumulate, it is picked up and removed by tiny lymphatic vessels and returned to the blood. As soon as the interstitial fluid enters the lymph capillaries, it's called lymph. Returning the fluid to the blood prevents edema. Again, returning the fluid to the blood prevents edema and helps to maintain normal blood pressure and volume. In addition to water, the lymph contains solutes derived from two sources. We have substances in plasma and substances derived from the cells, such as the hormones. So second function would be fat absorption. So the lymphatic system absorbs fats and other substances from the digestive tract. Lymphatic vessels called lacteals absorbs fats from the digestive tract. The lymph passing through the lacteals is milky in appearance due to the fat content. So you can correlate with this one in your CC, the chyle. And then the lymph and the lymph nodes serve as a massive network of vessels that function in filtration and trapping of microbe and other foreign substances. That's why it also has this function for defense. Again, filtration of the lymph by the lymph nodes and from blood by the spleen. So what filters the blood? It is the spleen. What filters the lymph? It is the lymph nodes. So the lymphatic capillaries and vessels. So the lymphatic system does not circulate fluid to and fro. So it's not like this. It's not back and forth, but it is a one direction movement. So one direction because of the presence of the valves in the lymphatic vessels. Again, the lymphatic system does not circulate fluid to and through the tissues. Instead, the lymphatic system carries fluid in one direction, from the tissues to the circulatory system. So, tissues to the circulatory system. The reason for this is that the lymphatic vessels are having valves. See, and the three factors that cause compression of lymphatic vessels: contraction of surrounding skeletal muscle during injury. This compression of the lymphatic vessels is, you might ask me, why is it beneficial or important? Because we can realize or see that in this example. Haven't you wondered why coaches or doctors will often urge athletes to exercise when they have a cold or other infection? What is the immunological reasoning behind this sage advice? Well, the contraction of your muscles, skeletal muscle and the smooth muscles, become the pump that helps the fluid get around your body. So the pumping action, it serves as the pump if there is contraction of the skeletal muscles. Exercise can help the lymphatic system flow more effectively and potentially help prevent infections and other diseases. So better stop being a couch potato and start to jog today. So that's it. Next, we will be having the discussion on the organs of the lymphoid system. Divided largely into two, we have the primary lymphoid organs, and we also have the secondary lymphoid organs. So primary lymphoid organs are also known as generative organs or generative, up to you how you will read it, while the secondary lymphoid organs are known as peripheral lymphoid organs. The difference between PLOs and the SLOs are the following. Primary lymphoid organs allow lymphoid stem cells to proliferate. Again, proliferation. They allow differentiation and maturation, while secondary lymphoid organs encourage functional lymphoid cells to grow. In addition, primary lymphoid organs contains only T cells or B cells, while secondary lymphoid organs contains T cells and B cells respectively. So again, the primary lymphoid organs and the secondary lymphoid organs constitute the lymphoid system or our immune system. You might also wonder, what do you mean by MALT or MALT? So MALT is a general term used to describe any immune structure found in mucosal tissue sites. More specific types of MALT include GALT, gut-associated lymphoid tissue. We have the NALT, or nasal-associated lymphoid tissue, and the bronchus-associated lymphoid tissue. So GALT, NALT, BALT. So we have all those. MALT may or may not have associated afferent or efferent lymphatics. Tonsils, adenoids, and the appendix are also considered a part of the MALT. And Peyer's patches are a type of gulls around the small intestine. So for the primary lymphatic organs, it is where lymphocytes are formed and mature. Again, maturation, differentiation, proliferation. And they give or they endow or provide an environment for stem cells to divide and mature into B and T. And there are two primary lymphoid organs, of course, the red, take note, the red bone marrow and the yellow bone marrow. And then also red bone marrow and the thymus. So only two for the primary lymphoid organs. And again, all lymphocytes arise from the multipotential, pluripotential hematopoietic stem cells that appear generally or initially in the yolk sac of the developing embryo and later found in the fetal liver. So it started in the yolk sac and then in the fetal liver. And later, when the infant is born, the bone marrow assumes this role. And the bone marrow, by the way, pluripotential means a cell that is capable of differentiating into cells of any type of tissue except placental tissue. And our pluripotential hematopoietic stem cells have CD34 positive markers on their cell surfaces. So again, please take note that the red bone marrow is the main source of hematopoietic stem cells. which again, hematopoietic pluripotential, meaning they can develop into any cell. Like here, if we are talking about pluripotential hematopoietic stem cells, they can become red blood cells, erythrocytes, granulocytes, monocytes, platelets, and lymphocytes. TB and NK cells arise from a common precursor known as the CLP. So this one, the CLP, common lymphoid precursor. So they will give rise to all cells, the T, the NK cells, B cells, and the DCs. Well, the common myeloid precursors will give rise to the granulocytes. We have, for you to easily remember the granulocytes, we have Ben, so basophil, eosinophil, and the neutrophils. So that's it. And please remember that the bone marrow is the center for antigen-independent lymphopoiesis. So independent, regardless of the presence or absence of antigens, the bone marrow will do or perform lymphopoiesis. So lymphocyte stem cells are released from the marrow and travel to additional primary lymphoid organs where further maturation takes place. And then one subset goes to the thymus and develop into T cells. In humans, B cell maturation takes place in the bone marrow itself. So that's it. And this one is the diagram of hematopoiesis. We have hematopoietic stem cells, again, which have CD34 positive cell markers on their surfaces. These cells are self-renewing. When they proliferate, at least some of their daughter cells remain as hematopoietic stem cells, so the pool of the stem cells does not become depleted. The process of the development of different blood cells from HSCs to mature cells is called, again, hematopoiesis for neurohematology. Lymphocytes are the cornerstone of the adaptive immune system. Okay? Commonly known as the WBCs, they derive from common lymphoid progenitors. So this one, lymphoid stem cell, common lymphoid precursor. This one is the common myeloid precursor. So please take note where they came from. So that's it. Next, we have the thymus. Thymus, it is a small, flat, bilobed organ found in the thorax or chest cavity or the mediastinum. And then at birth, it weighs approximately about 30 grams. And at peaks at puberty. First, they thought that early in life, the thymus produces enough virgin, take note, virgin T lymphocytes to seed the entire immune system. However, there is this new evidence indicating or suggesting that although the thymus diminishes in size, it is still capable of producing T lymphocytes until the 5th or the 6th decade of life, and some books are saying 4th decade of life. My question is, can we say that a person survived without the thymus? A person can live without their thymus gland, but the effects of not having a thymus depend on how old the person is. So how old? So haven't you wondered why I have asked that question? Because how would I say? The thymus, please remember that it atrophies and is replaced by adipocytes at the 4th decade of life. Losing a thymus gland for some reason, like having cancer or accident, would not be an immediate death sentence, but it would put a major deficiency into the working of the immune system. That would be especially traumatic at a young age. So it's a problem if you have lost your thymus at a young age. When the immune system is developing and the repertoire of T cells is expanding. With age, the loss would be less and less severe actually. The thymus, again, as what I have said earlier, devolves or involutes with age anyway, and in some point it might not matter that much. The memory T cells can survive for decades, so a person probably could do without a thymus gland, can live for the last several decades of life without noticing any significant effects. So that's it with the thymus. And let's go to your secondary lymphoid organs. And now talking about lymphopoiesis or reproduction of lymphocytes. It happens in the secondary tissue, but this is strictly dependent on antigenic stimulation. So this one is really dependent on the presence of antigens. While formation of lymphocytes in the bone marrow is antigen or antigen independent. again. So in the secondary lymphoid organs, it really requires or is dependent on antigenic stimulation. But in the red bone marrow, it is the center of antigen-independent lymphopoiesis. And most naive or resting lymphocytes die after leaving the PLO, the primary lymphoid organs, unless activated by the presence of a foreign antigen. What does naive means? Naive T cell or TH0 cell is a T cell that has differentiated in the thymus and successfully have undergone the positive and negative processes of central selection in the thymus. A naive T cell is considered immature and, unlike activated or memory T cells, has not encountered its cognate antigen within the periphery. In short, naive, no previous exposure of an antigen, or it doesn't have an experience. So it is in the second process that gives rise to long-lived memory cells and shorter-lived effector cells that are responsible for the generation of the immune response. And now let's go to the spleen. It is the largest secondary lymphoid organ. It filters the blood instead of the lymph, because what filters the lymph are the lymph nodes. And then this is the graveyard site for erythrocytes, because it is involved in the destruction of worn out red blood cells. And somehow the spleen serves as a blood reservoir. And then it has is an organ on the left side of the peritoneal cavity and has both immunological and non-immunological functions, composed of two types of tissues called the red pulp and the white pulp. So we have the red and the white pulp. So again, what are those? Two types of tissues that constitute the spleen. The white pulp is a part of the immune system and performs the same function as lymph nodes, but instead of surveying lymph, the white pulp surveys blood for microbial antigens, while the red pulp is associated with the veins. It's a fibrous network filled with macrophages and RBCs. So again, FYI, it is the largest single mass of lymphatic tissue in the body, the spleen, this one. And then within the red pulp, three functions of the spleen are found. Macrophages remove ruptured, worn out, defective erythrocytes, platelet storage, and hemopoiesis. Next, we also have the lymph nodes. Lymph nodes, they are located along the lymphatic ducts and are about 600 plus bean-shaped structures. And usually they occur in groups in the mammary, axillae, and groin. And what's the function of the lymph nodes? Mga lusay, say gamit. So they are the central collecting points for lymph. So the point is, lymph node, think of a filter or filtration. Try to filter the antigens. If you have antigens there, so it will be filtered. That's the time you can know or you can tell if there is trapping or filtration of these antigens when the lymph nodes are swollen or inflamed. Swollen lymph nodes usually occur as a result of infection from bacteria or viruses, and rarely, swollen lymph nodes are caused by cancer. Swollen, inflamed lymph nodes are a sign that they're working hard. More immune cells may be going there and more wastes could be building up. They function as filters, again, filtration, filter, trapping of the microbes. So that's for the function. Common areas where you might notice swollen lymph nodes include your neck, under your chin, in your armpits, and in your groin. And here, this is an example on cross-section of your lymph node. B cells congregate in the lymphoid follicles, also known as the B cell corona, and the marginal zone surrounds the B cell follicle and contains large amounts of marginal zone macrophages and disease. We also have tonsils. Tonsils, they are a large group of lymphatic nodules and diffuse lymphatic tissue deep within the Pharynx. They provide protection against bacteria and other potentially harmful material entering the pharynx from the nasal or oral cavities, the Waldear's ring. And then we have three types. We have the palatine, pharyngeal, and the lingual. And usually, we have the commonly we refer the palatine tonsils as the tonsils. Pharyngeal tonsils commonly referred to as the adenoid, and then lingual tonsils infected less often than the other tonsils because these two are commonly infected, the palatine and the pharyngeal. The clinical correlate for this one would be red and swollen tonsil, sometimes with white patches or streaks of pus, indicates strep throat. Strep, familiar? Strep throat. It is caused by infection with a bacterium known as Streptococcus pyogenes, also called Group A Streptococcus. It can complicate into rheumatic fever. Rheumatic fever, which is a serious inflammatory condition that can affect the heart, joints, and nervous system and skin. Other secondary lymphoid organs, we have the cold molt and the appendix. Here, macrophages and lymphocytes are localized as some of the main points of entry for foreign organisms. So the one that you should not forget will be this one, Bayer's patches, representing a specialized type of molt, and they are located at the lower ileum and intestinal tract. Next, we have the an overview of what really the immune system is. This classical view of the immune system, the self versus non-self. The hallmark of adaptive immunity, which requires engagement of innate immunity, is an ability to discriminate between self and non-self, and eventually between pathogen and symbiont, as well as peripheral control mechanisms maintaining immunological health and appropriate responses. Loss of control mechanisms and or regulation of either the adaptive or innate immune system lead to autoimmunity and autoinflammation. So we have this one, self, anything that is coming from the host genome and the rearrangement of host genes, cells, fluids, and anything else. Non-self refers to anything physically outside the host, whether an organism or a toxin. When foreign objects or damaged host cells are detected by the immune system, an immune response occurs. And immune responses occur or happen at different levels. We have the primary immune response and the secondary immune response. So here, it is showing an overlap of the innate and the adaptive immunity, because we cannot really say that what functions first is the innate and then followed by the innate and the adaptive. No, they work in an overlapping manner. There is really a crosstalk or interplay between the two types of immunity. The innate responses call the adaptive immune responses into play, and both work together to eliminate the pathogens. And please remember, natural killer T cells and gamma delta T cells are part of both the innate and adaptive immune response. The adaptive immunity is initiated when an innate immune response fails to eliminate a new infection, and antigen and activated antigen presenting cells are delivered to the draining lymph nodes. So here, this is a good summary of the innate or natural immunity, or inborn or non-specific immunity. This is represented by the primary lines of defenses of our body, the mucous membranes. Number one, of course, is the intact unbroken skin, and it is said that the innate or natural immunity really evolved earlier than the, of course, adaptive. This one is non-specific, always present. When you were born, we were born, it's there already. Thank you, mother, for passing those antibodies to us starting at the third trimester, or 7th month of pregnancy until birth. And this one is immediately available. Could be physical, biochemical, mechanical, and mechanism does not alter. or repeated exposure to any specific antigen. So the response is just the same to microbe or to anything. The innate immunity does not have a memory, so this is a bit a problem because is only present in your acquired adaptive immunity. So it's there. So the thing is, it cannot recall a previous contact with a foreign molecule. The components of the innate immunity are, we have the first line and the second line. So physical, chemical barriers of the skin and mucous membrane, second line, antimicrobial substances, NK cells, phagocytes. Take note, inflammation and fever. So we cannot really say that you don't like having fever, right? Of course, who would want to have one? But fever is not really that bad, and inflammation is really not that bad either. But the thing is, it has to be an acute inflammation so that we can say it's good, because if inflammation tends to become chronic because it's being prolonged with the presence of neutrophils, then that's the time we can say that it can lead possibly to autoimmune diseases. Innate immune responses represent immunity's early warning system, so early, and are designed to prevent microbes from gaining access into the body and to help eliminate those that do gain access. So here, the first line of defense is composed of structural barriers, really in the form of the physical barriers. We have the intact skin, mucous membranes, the cilia, cough reflexes, which are really needed. So don't stop or try to stop yourself from coughing or sneezing. And then we also have secretions. Secretions also have their functions. So it's not bad if you will sweat or if you will cry, if you're showering your saliva and you've got mucus in your nose, the bugs. And then also the very low pH of the vagina and the stomach. We require the pH there to be very acidic, not very, but it has to be acidic in the vagina and in the stomach. It has to be like pH 1, gastric, I think. And then again, first and foremost, it is the unbroken or intact skin and the mucosal membrane surfaces that are really protecting us. And to understand how important a role these play, one has only to consider how vulnerable victims of severe burns are to infection. Not only does the skin serve as a major structural barrier, but also the presence of several secretions discourages the growth of microorganisms. Take, for example, the lactic acid in sweat. For instance, in the fatty acids from the sebaceous glands, maintain the skin at a pH of 5.6. This acid pH keep most microorganisms from growing. The flushing action of urine plus its slight acidity, ayog tilawi, joke, and helps to remove many potential pathogens from the genitourinary tract. Lactic acid production in the female genital tract keeps the vagina at a pH of 5. So girls, don't try to clean your vaginas that much. They can clean themselves. By themselves, they can maintain that pH. So if by the time that the pH really can change, though, there might be possible pathogenic invasion. And on the moreover, in the digestive tract, the acidity of the stomach, which is due to the production of HCl or hydrochloric acid in the stomach, HCl, keeps the pH at as low as 1, yeah, I'm right, and serves to halt microbial growth. Lysozyme is an enzyme found in many secretions such as tears and saliva. So tears and saliva both have lysozyme. Take note, zyme, enzyme, and this one is lysis. Especially for gram-positive organisms. So these lysozymes are effective against gram-positive microorganisms. And in many locations of the body, there is normal flora that often keeps pathogens from establishing themselves in these areas. This phenomenon is known as competitive exclusion. So much for the first line of defense. For the second line of defense, we have the internal components. the cellular and the humoral, and acute inflammatory reaction under the second line of defense under innate natural immunity. So it's not under the first line, but the second line of defense under innate or natural immunity, your acute inflammatory reaction or infection. So it's easy for you to remember cellular CT because T cells are involved here, and humoral fluid. So it's we also have complement, the complement and the cytokines, chemokines, interferons, and interleukins. So let's dig deeper. The skin. The good thing, the skin's lining epithelium is keratinized stratified squamous epithelium, and this periodic shedding of the epidermal cells help remove microbes at the skin surface, and bacteria rarely penetrate the intact surface of healthy epidermis. If the surface is broken by cuts, burns, or punctures, however, pathogens can penetrate the epidermis and invade the underlying tissues or adjacent tissues. Next, we have the mucous membranes. So mucus is on functions in lubrication, and it moistens the cavity surface. It is slightly viscous in a sense so that it can trap the microbes and the foreign substances. We have mucus in our nasal mucous membranes, and the hairs are coated with mucus that trap and filter microbes. You also have the cilia that has a wave-like action to propel the antigens away, and also coughing and sneezing to accelerate the movement of mucus and its entrapped pathogens out of the body, and swallowing too. Mucus sends pathogens to the stomach where the gastric juice destroys them. That's why it's okay to swallow your mucus. After all, the gastric juice will eventually kill the antigens that are trapped within them. Next, tears and saliva. Again, to summarize, we've got IgA and the lysozymes. Tears functions in it has a washing action that can dilute the microbes and keep them from settling in the eyes, and contains lysozymes. Lysozymes, again, capable of breaking down the cell walls of certain bacteria. And talking about the other one, the saliva, it is produced by the salivary glands, washes microbes through the surfaces of the teeth and from the mucous membranes of the mouth, much as tears wash the eyes. The flow of saliva reduces colonization of the mouth by microbes. Urine also cleanses the urethra and retards microbial colonization of the urinary system. So urine, vaginal secretions move microbes out of the body in females. Defecation and vomiting also expel microbes. So it's okay to defecate and vomit if you are having food poisoning. Next, second line of defense, internal defenses. We have antimicrobial substances in the form of interferons, complement system, and iron-binding proteins. What are the functions of the interferons? They are secreted glycoproteins that are produced by the cells in response to, take note, virus infection. When the cells are virally infected. Another stimuli and induce antiviral state in cells bearing interferon receptors. In this way, interferons restrict virus replication and spread before an adaptive immune response is developed. While complement was discovered many years ago and is component of normal plasma that augments the opsonization of bacteria by antibodies that allows antibodies to kill some bacteria. This activity was said to complement the antibacterial activity of antibody, hence the name. So that's for your complement. But the endpoint really for complement activation would be cytolysis, aside from phagocytosis and inflammation. We also have antimicrobial proteins. In our sweat glands, we have dermicidin, we have defensins and cathelicidins. Secreted by macrophages and in the epithelia and the neutrophils, and we have thrombocytin in our platelets. So this one, why I specifically tried to discuss natural killer cells and phagocytes in one slide, because NK cells function as important sentinels of the immune system. They work as primary responders and alerts the host to the presence of infectious organisms. These cells represent a subset of cytotoxic lymphocytes these cells are able to recognize and lyse tumor cells, so lysis of tumor cells. And then these cells lack the membrane molecules that identify B and T cells, but they have the ability to kill a wide variety of infected body cells and tumor cells. Tumor cells, virally infected cells too. And they attack any body cells that display abnormal or unusual plasma membrane proteins. The binding of your NK cell to the target cell or any infected human cell will lead to the release of granules. These granules have toxic substances coming from the NK cells. Granules in the form of perforin. Perforin insert into the plasma membrane of the cell, creating perforation. So meaning na ay buslot, iyan ang buslutan, creating holes or perforations. What will happen? The ECF of the cell will get outside. Hence, it will lead to cell rupture or the cell will burst. And this process is known as cytolysis. Cytolysis. Okay, so cytolysis, what cell is capable of doing this? The NK cells. Other granules of NK cells release granzymes. So perforins, granzymes, correlate them to NK cells. Granzymes are protein digesting enzymes that induce the target cell to undergo apoptosis or programmed cell death. Okay? Programmed cell death or apoptosis. However, you might think cell death is always pathologic. It's not. This one is the physiologic type of cell death. We also have phagocytes. The most active phagocytes, FYI, are the neutrophils and the monocytes. There are special cells that perform phagocytosis, phagian to eat, right? And preparing to eat, because we have oosis, the process. So it is the ingestion, engulfment of microbes, and other particles such as cellular debris. We have two major types of phagocytes. We have the neutrophils and the macrophages. So neutrophils, they are the first responders, but the last to arrive, but won't leave unless the infection and inflammation is clear, the macrophages. So again, when an infection happens, neutrophils and monocytes migrate to the infected area. It could be through chemotaxis or diapedesis for the neutrophils. So during this migration, the monocytes enlarge and develop into actively phagocytic macrophages called wandering macrophages, because we have two types of macrophages, the fixed macrophages, and we also have the wandering macrophages. Fixed macrophages stand guard in specific tissues. They are localized in specific tissues. Examples would be in our connective tissues, we have histiocytes. In our liver, we have the Kupffer cells. And in the lungs, we have the dust cells or the alveolar macrophages. And in the CNS or in the nervous system, we have the microglia. And in addition to being an innate defense mechanism, phagocytosis plays a vital role in adaptive immunity. And please, again, neutrophils are abundant in the blood and essential in acute inflammation, because neutrophils, they are the first immune cells to arrive at the site of infection. And macrophages are tissue-resident cells that act as an initial defense mechanism and serve to activate the adaptive immune response. But the thing for you not to forget, these two types of phagocytes are like boys, like someone who will arrive that early, that will be your neutrophils if you are in pain. But the bad thing, it's the first to arrive and the first to leave. However, it's alright really to wait, because macrophages, likened to the macrophages, yes, they arrive late, but they will remain with you until you are okay. So choose wisely. Next, we have the major types of phagocytes again. No, sorry. We have the major types of phagocytes, neutrophils, and macrophages. But before talking about that, monocytes in the tissues, macrophages. No, monocytes in the blood, macrophages in the tissues. I mean the location. Okay, so let's go to phagocytosis. We have the steps. Chemotaxis starts with chemotaxis. It is the movement in response to a chemical stimulation or the presence of these chemical attractants. Next, followed by adherence or attachment to a microbe. This is the microbe. This is the microbe. And then that microbe will be ingested, engulfed. There is ingested by, of course, the phagocyte. And then ingestion is the engulfment of the pathogen with the pseudopodia. This is the pseudopodia, right? Can you see it? And then followed by digestion. Digestion wherein there is phagous cell maturation, and then followed by elimination wherein phagocytes eliminate remaining pieces remaining pieces of microbe via exocytosis. So, however, not all are released because we have indigestible materials known as residual bodies, and you can appreciate this one in your histopathology. So we also have inflammation. Inflammation is the immune system's response to harmful stimuli, such as not only pathogens, but also damaged cells, toxic compounds, or irradiation, and acts by removing injurious stimuli and initiating the healing process. See, inflammation is okay. And then, inflammation is therefore a defense mechanism that is, of course, vital to health. And inflammation is a vital part of the immune system's response to injury and infection. It is the body's way of signaling the immune system to heal and to repair damaged tissues as well as defend itself against foreign invaders such as viruses and bacteria. Without inflammation as a physiological response, wounds would fester and infections could become really deadly. However, if the inflammatory process goes on for too long, or if the inflammatory response occurs in places where it is not needed, then it can become problematic. Chronic inflammation has been linked to certain diseases such as heart disease, stroke, and may also lead to autoimmune disorders such as RA and lupus. But a healthy diet and lifestyle can keep inflammation under control. So, clinically, acute inflammation is characterized by 5 cardinal signs. Some books would say only 4, others would say 3, but now we have 5 already. So, why 5 cardinal signs? Cardinal signs common would mean common signs and symptoms. So, let's start with rubor. What is rubor? Redness. This one. We have calor. Where is calor? So heat or increased heat. We have tumor, swelling, and dolor, pain. Where is pain? This one. Pain and functionalesa, which is loss of function. Well, the first 4 of the signs and symptoms of the 5 cardinal signs of inflammation was described by Celsus. First 4 described by Celsus. However, loss of function was later added by Virchow. So, are we okay with that? Next, we have fever or pyrexia. It is an elevation of the body temperature caused by a cytokine-induced upward displacement of the set point of the hypothalamic thermoregulatory center, the hypothalamus, in short, hypothalamus. The purpose of fever isn't completely understood, but small elevations in body temperature appear to enhance immune function and inhibit pathogen growth. So, it's on the control of the growth of the pathogens. Many bacterial toxins elevate body temperature, sadly, sometimes by triggering releasing of fever causing this one, cytokines, again, IL-1. But it's not only IL-1, we have IL-1, 6, and the tumor necrosis factor TNF. See, we have interferons too. Next, we have acquired or adaptive immunity. So it supplements protection provided by innate immunity, and there is later evolutionary development and is seen only in vertebrates. This one, if you will ask me how can I summarize the acquired adaptive immunity into two words, I would say specificity and memory. Again, specificity and memory. Specific recognition, and there is the selective elimination because of it can recall the previous exposure of an antigen, pathogen, or a microbe. So let's start with specific or specialized. How can we acquire adaptive immunity? Acquired because of the initial contact. You need to have the primary contact with the foreign substance, which triggers synthesis of specialized antibody proteins resulting in reactivity to that particular foreign substance. So for you to have memory on a particular antigen, you need to be exposed to that antigen. Understand? Next, memory. Response improves with each successive encounter. Are you like this whenever you're studying? If you can read something again and again, there is an improvement with the memory? Yes or no? Okay. Next, with the same pathogen. So it remembers the infectious agent and can prevent it from causing disease later. So quite amazing. Immunity to withstand and resist subsequent exposure to the same foreign substance is acquired. So that's your acquired or adaptive immunity. So again, this one, adaptive, acquired, specific, and refers to defenses that involve specific recognition of a microbe brought about by the presence of antibodies. Okay? Is based on a specific response to a specific microbe. That is, it adapts or adjusts to handle a specific microbe. Unlike innate immunity, adaptive immunity is slower to respond. Again, remember, innate, always there, and the response is rapid. But this one, though the response is slow, but surely it is really specific, and it can really remember what antigen that one is facing, your immune system is facing. Adaptive immunity involves lymphocytes, B cells, T and B cells. So we have, if we are talking about third line of defenses, not defenses, okay? I have seen some students having a wrong spelling with this one. It's defense, okay? Not defenses, okay? Next. So we have the internal components, cellular, CT, because we have the lymphocytes, T lymphocytes, we have the TH, TC, and the, what do you mean by TH? Helper, TC, cytotoxic. And we have B cells, the B memory cells, and the plasma cells. Well, plasma cells are mature B cells. They have matured, or they were able to reach maturation upon the exposure or the first encounter of the antigen. So in the presence of the antigen, B cells become mature cells known as plasma cells. Plasma cells produce antibodies. They are the only cells in the body capable of secreting, synthesizing gamma globulins, immunoglobulins, or antibodies. Next, we have the APCs, or antigen presenting cells. Why, you might wonder, what's the use of such cells? These cells are responsible of capturing the antigens, and after capturing, they will digest it, and after digesting, the the antigen now would be fragmented, and those fragments of the antigen will be presented by the APCs to the T cells by using MHCs, or your HLA molecules, or your major histocompatibility complex molecules. Again, APCs are useful because they are needed by the T cells to recognize antigens. Why am I trying to emphasize this one? T cells cannot recognize intact antigens. Intact antigens are recognized by B cells but not by T cells. So T cells, like somehow, like so weak, cannot recognize intact antigens. So they need other cells to present the antigens for them. So in short, kutihan, like tanga nga kutihan kay di pa jud kay ila kinahanglan pa jud na yung present, di ba, nakagets? And then next, We have humoral. So humoral always think of, it should think of antibodies. And we have the complement in the, well, we have the three types of complement. We have classical, but here we only have the classical pathway, and we also have the cytokines. So cellular immunity, well, some books would say cells attacking cells, cell-to-cell contact. See, cytotoxic T cells directly attack invading antigens. Cytotoxic T cells are known as your CD8 T cells, CD8 positive, because these are T cells. These are cells. For example, this is a CD8 positive T cell. The marker in the cell surface of your T cell is CD8. That's why you say they are CD8 positive T cells. But if you are talking about helper T cells have CD4 markers on their cell surfaces. So we would say that helper T cells, helper T cells, CD4 positive T lymphocytes. So that's this is a way of CDs, or clusters of differentiation. This is a way for us to tell or to know what particular cell is that, specifically for our lymphocytes, for our leukocytes, for WBCs, for us to differentiate the WBCs, the granulocytes, the lymphocytes, like that. Class, remember, for a while, not the eraser. Okay, so that we can differentiate the lymphocytes, we need to have them, we need to know their cell surface markers. And you might ask me, how are we able to do that, or classify them, or tell that this cell have this particular surface marker on its surface? It is brought about by immunophenotyping, by flow cytometry. Okay? Don't worry, we will have a separate discussion of that one in our flow cytometric studies later in module 8 or 9, I'm not sure. And then next, going back to the topic, here, cellular immunity, cellular, so lymphocytes, very effective, highly potently effective against intracellular pathogens. If we are talking about T cells, they are for intracellular, like viruses, tumors. But if we are talking about extracellular, what cells are responsible, effector cells responsible for trying to combat such antigens, it would be the B cells. B cells are useful in extracellular antigens or pathogens, while your T cells are useful for your intracellular. And another thing is, for B cells can recognize intact antigens, while T cells require those antigens to be fragmented. Okay, fragments. Next, going back to your cellular immunity, cellular immunity is really potent against intracellular pathogens, and then cancers, neoplastic cells, and transplants. So whenever there is organ transplantation rejection, always blame the T cells for that. The T lymphocytes are the main culprits why there is rejection, which later you will understand in our organ transplantation discussion. Next would be, we also have the antibody-mediated immunity, antibody-mediated, humoral-mediated immunity. The same, all the types of the processes in this type of immunity are mediated by the B cells. So here, antibodies are at play. Antibodies. So it involves antibodies that bind to antigens in body humors or fluids such as blood and lymph, and also it is also known as your humoral immunity. Next, we have clonal selection. What do you mean by that? This phrase is common in immunology, but hardly understood. To simplify, clonal selection is the process by which a lymphocyte divides or proliferates, and it differentiates to more specialized cells. In response, clonal selection happens only in response to the presence of a specific antigen. So if there's no antigen, there's no clonal selection. You get the point? So What is the result of clonal selection? Is the formation of clones. Clones. What do you mean by clones? Clones have they have similar, or yeah, very similar. They have the same antigen receptors. The same antigen receptors. So, for example, this clone and the other clone, both of them can tell that this is the antigen, that this antigen, for example, is the dog antigen. Like they can recognize the same antigens, that they can recognize the same specific antigen as the original lymphocyte. That's the meaning of being a clone or clones. Location, where can we find the clones? Not in the primary or generative lymphoid organs, but in our SLOs or secondary lymphoid organs. So we have two types of clones, of course: effector cells and memory cells. Effector cells, those that really carry out the actual combat, carry out immune responses. Immune responses in the form of destruction, elimination, inactivation, opsonization, or somehow neutralization of the antigen. So examples are always take note, you should include active helper T cells, active cytotoxic cells, or plasma cells. We also have other clones in the form of memory cell clones: memory helper, memory cytotoxic, and memory B cells. You might wonder, Miss, how long can those memory cells remain? Well, memory B cells, memory B cell, memory T cells can remain like decades, a decade, 10 years, or the entire lifetime of the person. Next, antigens and antigen receptors. Two important characteristics of antigens: immunogenicity and reactivity. Immunogenic and reactive. What do you mean by immunogenicity? Really, that it can elicit an immune response, and the response is seen in the production or synthesis of antibodies. That's why the term, the line, sorry, all immunogens are antigens, but not all antigens are immunogens. Because yes, immunogen meaning it can really cause the it can really activate the immune system, specifically the humoral or the B cell mediated immunity. Okay, the antibody mediated immunity under your acquired adaptive. However, it can become, yes, we can say it's antigenic, but not all antigens can really induce an immune response. Okay. See, immunogenicity is the ability to provoke an immune response by stimulating the production of specific antibodies plus the proliferation of specific T lymphocytes, or both. The term antigen derives from its function as an antibody generator. Take note, antigen. See, anti body generator or generating. So in the presence of your antigen, antibodies are generated. See, there's a sense in that. Next, reactivity is the ability to the end of the antigen to react specifically with the antibodies or cells it provokes. So there is this specificity. Why? Because the FAB, antigen binding fragment of your antibody, will only bind to the specific epitope of an antigen. Typically, just certain small parts of large antigen molecules act as triggers for immune responses. Yes, because it is not the entire antigen that will become immunogenic to our immune system. No, it will be immunogenicity is observed, and specificity will only be observed through the presence of the epitopes or antigenic determinants. So these small parts are called again antigenic determinants or epitopes. These epitopes induce the production of antibodies and not the whole part of the antigen. So example would be in your gram-negative, I think will be the LPS. For gram-negative bacteria, it's not the entire bacterium that is antigenic. It's just a part of it in its cell wall, or some in the flagellar H antigen, like that one. Next, we have chemical nature of antigens. So how can we describe what is an antigen? So it has to be large, it has to be complex, the molecule has to be complex, and they have to be, most proteins are really, of course, antigenic. They should be proteins. However, nucleic acids, lipoproteins, glycoproteins can also act as antigens. And we also have the term complete antigens. What do you mean by complete? Because they are really large. Molecular weight will be 10,000 daltons or more. But large molecules that have simple repeating subunits are not antigenic. Take note, even molecules that are large but their structure are simple, they are not antigenic. Again, large molecules that have simple repeating subunit simplicity, even though if you are large and you think you're an antigen but you're just a simple person, you're not antigenic. You have to be something like unique, and you have to be really, like, how to say, complex for you to become an antigen. And a smaller substance that has reactivity but lacks immunogenicity is a hapten. Hapten meaning to grasp, to hold, right? Hapten, because normally it is small. Normally it is not, it doesn't elicit immunogenicity. However, once this hapten can bind to a carrier molecule or is attached to a larger carrier molecule, that's the time that this hapten turns to become or becomes an antigen. That's why hapten-stimulated immune responses are responsible for some allergic reactions to drugs and other substances in the environment. Take, for example, penicillin, okay, can become a hapten in some instances. That's why some people are allergic to penicillin, right? Because the drug molecule of your penicillin, the penicillin drug molecule, was able to attach to albumin, and somehow the immune system recognized it as an antigen. So a sad thing. So you might, I would just like to make this one a point that not all functions of the immune system is really beneficial. It's not always desirable. Sometimes the effects of our immune system will be undesirable, and you can observe such undesirable effects if you are having a blood transfusion rejection or organ transplantation reaction, or you're having, like, for example, RH incompatibility, hemolytic disease of the newborn. That's the time that you can say, oh, our immune systems are not needed here. They're trying to do this, but the thing is, instead of giving us the good thing, they are adding up to the burden. Don't worry, we will be having a separate lecture for that too. Now we will be having the major histocompatibility complex antigens. This one will just be a review because Ma'am Pam or me will discuss this one further. So what are MHCs or HLAs? As what I have said earlier, MHCs are utilized by your, how to say, antigen presenting cells. And they function in your acquired adaptive immunity, specifically under cellular mediated immunity. The CD4 and CD8 lymphocytes are involved here. So going back, we have major histocompatibility complex antigens. These are located in the plasma membrane of body cells. Plasma membrane, not nuclear, not cytoplasmic, but in the plasma membrane, okay? These transmembrane glycoproteins are also called the human leukocyte antigens because they were first identified on WBCs or white blood cells. And unless you have an identical twin, your MHC antigens are unique. So in this way, if someone is saying that you're not unique, you really are. Not unless you have an identical twin in the context of your MHC antigen expression. Nga naman, ngano di ay, ang twins are may pwede good direct na transplant, diba? Ngano kung ikaw na kay wala kay twin, matransplantan di ka, dili, diba? So that's it. That's the main point for that. Next, the normal function for the MHC antigens is to help the T cells recognize or to tell that an antigen is foreign or non-self or not self, okay? And this recognition, such a recognition is really essential as a first step in any adaptive immune response. And we have two types of MHC complex antigens. We have the class 1 MHC molecules and the class 2 MHC molecules. Class 1 MHC1 are built or they are found in the plasma membranes of all body cells except red blood cells, except RBCs, okay? So, nana siya tanan, basta wala lapot ang red blood cells. And we also have class 2. They appear on the surfaces of APCs. So APCs, we have DCs, dendritic cells, we have macrophages, and monocytes. Okay? And then in the scientific context, the function of your MHC molecules is to bind peptide fragments. Fragments coming from those antigens derived from those pathogens and display them on the cell surface for recognition by appropriate T cells. Okay? As they are being utilized by the APCs. Di pa masabtan karon no kay walay drawing sa gilang. Wala gyapon na drawing in the future. Joke bitaw eh. Nanana siya. Kung ako ay mag-lecture, hindi man murag mampam ni siya. Si mampam na lang siguro. Next, we have the cytokines. Cytokines, according to Stevens, they are chemical messengers. Messengers because they are regulators of host responses to infection, immune responses, inflammation, and trauma. Some cytokines act to make disease worse, yeah, sadly, pro-inflammatory, others serve to reduce inflammation and they also promote healing. So, funny, contradictory, some cytokines are pro-inflammation, ang uban kay anti, diba? So, up to the kind of signaling that is being activated when there is an infection, inflammation, or trauma. So, cytokines include chemokines. Diba? Dili na balihon, kay daghan jud masayop ana. Many would think that chemokines ang general then smaller ng cytokines. No, cytokines general under na niya ang chemokines, interferons, interleukins, lymphokines, and TNF. But generally not hormones or growth factors. And cytokines are produced, nagikan miss, by a broad range of cells including immune cells like macrophages, B lymphocytes, T lymphocytes, and mast cells. Asang mast cells sa tissues, diba? As well as endothelial cells, fibroblasts, and various stromal cells. So, what's your function miss? They act through cell surface receptors and especially important in the immune system because they modulate the balance between humoral and cell-based immune responses, and they regulate the maturation, growth, and responsiveness of particular cell populations. Masabtan na ninyo sa didaghan kayong cytokines, so wala ko ninyo memorize but be familiarized na lang. Okay, so kato siya, proliferation, pro-inflammatory or anti-inflammatory, depende na siya kung asa po siya na involved na kuan. So there are origin and functions. This is a list of your cytokines. So we have daghan jud kina siya class, honestly kani siya ang important ra ni giapil ni Stevens. Daghan gid kaay ni kung mag-masters mo no more di na maganahan mo mata joke. So IL1, IL2, IL4, 5 gamay lang na siya, at least kabaw mo na 1 2 3 4 5 so na mo check, di ba? We also have TNF, TGF beta, gamma interferon, alpha and beta interferons, perforins, perforins, granzymes, earlier, kinsa ganina sa NK cell man na, diba? Granzymes, perforin, apil na sa cytokines. Specifically, lymphocytes or other cells in the immune system really do, pero ang for you to easily remember, lymphocytes do release cytokines because they might accentuate or they can cause downregulation or upregulation of a particular immune response. That's with your cytokines. And so, now, kanisha, easy na tabalik. Acquired adaptive immunity further subdivided into active, passive, natural, active, natural, active, artificial, passive, natural, or passive, artificial immunity. This one is very easy. And you need to really memorize this one because surely it will come out in the board exam. So this one, it is possible to acquire adaptive immunity in 4 ways. So kanisha, you have your active natural, active artificial, passive natural, and passive artificial immunity. The terms natural and artificial refer to the method of exposure. So your active and your passive, so methods of exposure, how the person was exposed to the antigen. Natural exposure implies that contact with an antigen or antibody occurs as a part of everyday living and is not deliberate, meaning dili intentional. Artificial exposure, also called immunization, is a deliberate introduction of an antigen or antibody into the body, diba? The terms active and passive indicate whether or not an individual's immune system is directly responding to the antigen. When an individual is naturally or artificially exposed to an antigen, an adaptive immune system response can occur at that produces antibodies. This is called active immunity because the individual's own immune system is the cause of the immunity. Meaning getting active because the immune system is working, the immune system is producing antibodies. Nakasabot. Why? Can we say that acquired, adaptive, active, natural, artificial immunities are long-term types of immunities? Yes. Long-term, kani sila. However, your acquired, adaptive, passive, natural, artificial immunity, short-term lang yan because antibodies given there, the antibodies there are just transferred and those antibodies can be consumed up in the process. So that's it. Okay? So, question, going back. For example, passive immunity occurs when another person or an animal develops antibodies, and the antibodies are transferred to a non-immune individual. Again, passive immunity happens if one person or an animal develops antibodies and these antibodies are transferred to a non-immune individual. So nakaisa ka individual and you got the serum and you've got the antibodies in the serum transferred to another individual. That's your passive immunity. For example, infants can acquire antibodies from mother's milk or in utero. Yes, kan siya passive maternal transfer of antibodies to the baby, di ba? So kani natural. Bakit passive nga no? Is it the baby producing the antibodies? Is the baby's immune system producing the antibodies? No, it is coming from the mother. That's why passive. Nakasabot ang punto ha, di ba? And how long the immunity lasts differs for active and passive. Mo ni akong giingon earlier. Active immunity can persist for a few weeks to a lifetime. So, ang long term niya siya, few weeks to our lifetime, active immunity. Immunity can be long-lasting if enough B or T memory cells are produced and persist to respond to later antigen exposure. So, kana siya katuin kung a lifetime, di ba? Decades, 10 years to a lifetime. Passive immunity, on the other hand, is not long-lasting. This one, short-term, because the individual does not produce his or her own memory cells here. Dili man, hinatagar man, diba? Because active immunity can last longer than passive immunity, it is the preferred method. Again, because active immunity can last longer than passive, this is the preferred method. So, kani better. So, meaning magpasakit na lang jud mi para kuan, yes, natural inoculation, subclinical or clinical disease, naturally na-expose ka sa nature, okay na siya. Rather than gihatagan kag vaccine, di ay parihara bitaw kay active ang gyapon ng vaccine. Dili ko anti-vaxxer ha. Okay? Again, passive immunity is preferred. However, immediate protection is needed. However, when immediate protection is needed, active natural immunity class kay unsa man. Kung gadali ka, pag passive ka, diba hatagan kay kung kani mag-wait pa man mo produce imong immune system og antibodies. Next, we have, again, active natural immunity. So, kaniha, one by one. Acquired adaptive, active, natural immunity. So, this one is, happens through natural exposure to an antigen, such as a disease-causing microorganism, and can cause an individual's immune system to mount an adaptive immune response against the antigen and achieve active natural immunity. Because the individual is not immune during the first exposure, here, during the first exposure, dili pa man siya immune, he or she usually develops the symptoms of the disease. So, masakit dyan na siya. Interestingly, exposure to an antigen does not always produce symptoms. Many people exposed to for example, kanang mga exposed to poliomyelitis virus at an early age, have an immune system response and produce poliomyelitis antibodies, yet they do not exhibit any disease symptoms. Diba nice? Next, we have your acquired adaptive, active artificial immunity. So in an active artificial immunity, an antigen is deliberately introduced into an individual to stimulate the immune system. A best example with this one will be the process of vaccination. Kanang intentional good nga gi-introduce ang antigen ba, kanang vaccine day ma'am, unsa din na component ana. Antigens na sila, di ba? Dili na ang antibody gihatag, di ta magbinugo here. Okay? So, again, the process of vaccination and examples will be, also may example any, DTP injection for diphtheria, tetanus, and pertussis for your whooping cough, diba? Na kay MMR, katong, unsa pa man. And nasa kay OPV, familiar or oral polio vaccine, na siya. And the vaccine usually consists of a part of a microorganism. Pwede part lang sa microorganism. It could be live. But kung live na siya, mas severe ang side effects. So, mo gani dapat dead na lang, pwede attenuated or weakened or gi-alter through heat, aging, or chemical means sa vaccination. Sige lang, mag-lecture po tag vaccine, isa naka-module, last na siya. And then, Ma'am, nga naman siyang kuan, nga nung gi-alter ang antigen. The antigen has to be changed para so that it will stimulate an immune response. But take note, but will not cause the symptoms of the disease so that the person vaccinated will not contract the disease. Di siya masakit, di ba? Because active artificial immunity produces long-lasting immunity without disease symptoms. It is the preferred method of acquiring adaptive immunity. Acquiring adaptive immunity, unsagay the best method na siya kanisha. Active artificial through vaccination. So next, we will go to your passive immunity, passive natural, acquired adaptive, passive natural immunity. So, it happens results from the transfer of antibodies from a mother to a child. Ako nang gibalik ha, across the placenta before birth. And during her life, di ba, ang mama, the mother, has been exposed to many antigens. The more gani antigens na exposed ang mother, the better. Di ba? So, maghugawan mo dapat, joke. Either naturally or artificially, and she has antibodies against many of these antigens. These antibodies protect the mother and the developing fetus against disease. Some of the antibodies, IgG, can cross the placenta. Take note, IgG can cross the placenta and enter the fetal blood. Following birth, the antibodies provide protection for the very first few months of baby's life, lasting until the 6th month. Napanay antibody protection from the mother ang baby. Kabantay mo ika 7th month or 8th, kaya magsakit-sakit na siya. Diba? Or kung mo kabantay kay, wala siya mo kabantay. May pake, joke. Next is, eventually, the antibodies are broken down and the baby must rely on his or her own immune system. So, if the mother nurses her baby, kasi nurses, ma'am, kanang nursing, dili, nurses, nagpatutoy sa bata, antibodies, IgA, in the mother's milk may also provide some protection for the baby. So, aside sa placental transfer, napuke transfer through the breast milk. Kana siya. Next is passive artificial immunity. How can we achieve this one? Usually, it begins with vaccinating an animal such as pwede siya kabayo or horse. After the animal's immune system responds to the antigen, antibodies are removed from the animal and injected into the individual requiring immunity. So, in some cases, a human who has developed immunity through natural exposure or vaccination is used as a source of antibodies. So, nag-need sila og your passive artificial requires an animal or human as the source of the antibodies or gamma globulins, immunoglobulins. So, passive artificial immunity provides immediate protection. Take note, immediate protection kung gadali gyud ka. So, you will rely on this one, passive artificial immunity. because it provides immediate protection for the individual receiving the antibodies and is therefore preferred when time might not be able for the individual to develop his or her own immunity. Diba? Kanang gadali jud ka kay napaaka naskuan, diba? This technique provides only temporary, however, kanilagi siya short term. If the first two are long term, the kani siya natural artificial passive immunity short term na jud na siya. So this technique provides only temporary immunity, however, because the antibodies are used or eliminated by the recipient. Yung gamit, magud siya, mawala na siya. So, anti-serum is a general term. Anti-serum. Is a general term used for serum, which is plasma minus the clotting factors that contains antibodies responsible for passive artificial immunity. So, ang sige use good jud ma'am na antibodies, ma'am, anti-serum. Anti-sera are available against microorganisms that cause diseases such as measles, hepatitis, rabies, tetanus, diphtheria, and botulism. Kana siya, pwede yun na siya. And next, so easy na lang. Here, we have the comparison of innate and adaptive immunity. So memorize yun siya nga table, kay very important. So primary cells here, for your innate, always includes the phagocytes, right, kabantay? And if we are talking about antibody-mediated, ani man siya? One student in his or her Google form, katong sa things I haven't learned or I wanted to understand, kani siya, if you are confused between innate and adaptive, very easy. Under your adaptive immunity, you have your antibody-mediated and your cell-mediated immunity. But in your innate immunity, you have the first and the second lines of defenses. Ano na siya? So in your, we will go with this one by one. Okay, innate immunity, primary cells, kani. But for your antibody, kinsa man di mo produce antibodies ang plasma cells, which are mature B cells, di ba? We also have cellular mediated immunity. Ngano ganing mga cell mediated kay cells attacking cells, di ba? Cytotoxic example. So we have T cells here, T lymphocytes. Here, B cells for your antibody, cell mediated T cells. Para di mo makalimot funny kaayo ayun na lang, di mo kabawat sa akong mnemonic. Kanina lang CT, masayon, CT, cellular T cells. Origin of cells, galing. Saan galing lahat? Red bone marrow, of course. Miss, origin man, take note ha, kung yung maturation, I said origin. So kung origin, saan galing, di ba? The site of antigen independent unsa to siya, lymphopoiesis happens in the red bone marrow. Take note. But if ningon ko, if I'm saying maturation gani asa sila ni mature. So B cells, of course, mo mature. Bone marrow, B, di ba? For thymus, T cells, di ba? Sa yung lujukin na i-memory siya, makalimot pag yun. And then, location of mature cells, man o siya, miss, sa'yo na kayo na, lymphatic tissues, ubla, pa man din sa brain, later, regional, regional immune responses, because, I would say the brain, some cells cannot enter the brain like the immune cells. Later, ako na-explain. Primary secretory products, we have here in your innate immunity, histamine, we have your kinins or kinins, complement, very common. And for your antibody-mediated immunity, antibodies, of course, kasi yun. Kanina man siguro marong, di ba kaningon ko ganyan, as earlier I told you, cytokines are produced by lymphocytes. Majority, not all, but majority of the cytokines are produced by lymphocytes. Primary functions, we have inflammatory response and phagocytosis, inflammation under the Some books class would say complement still is under innate immunity and still under adaptive, pero kung simple lang yun, complement starts in innate immunity, functions largely in innate immunity. Pero napod na siya ang overlapping function here in adaptive. And then, what is important, you need to memorize this one. Extracellular B cells, intracellular, intact B cells, intact antigens, fragmented antigens T cells. Ano lang yun. And then, does hypersensitivity reactions happen? Yeah, in our cellular mediated immunity. In our cellular, this one is delayed, this one is immediate. So please memorize this table. Memorize. So the discussion for antibodies will be for MAMPAM. Antibody structure, please go over with this one because this is no longer my topic. And just please be familiarized with this table, a very important table. I love this table because you can really see the pentameric IgM, the dimeric IgA, na siya. And please be familiarized with the functions na lang, okay? So, as a whole, this is immunology and serology. So, the immune system, how the immune system is being organized. So, we have the physical barriers. Physical barriers, the skin and the mucous membranes, so correspond to our innate immune responses. Under innate, we have inflammation, cytokines, the interferons, we have the complement, NK cells, and under adaptive, we have the humoral for our B cells, cell-mediated for T cells, CD8, CD4, because these are lymphocytes. And for B lymphocytes, further, they have plasma. But our CD8 and CD4 also have memory cells, so we need to include that. So that's it for the first discussion of immunology and serology. I hope you have learned something or you have clarified some of the things that got you confused before. Thank you for listening, and I am hoping that you will still listen to my recordings for the next discussions. Good day. And please remember, courage doesn't always roar. Sometimes courage is the quiet voice at the end of the day saying, I will try again tomorrow. Thank you and God bless.